Good morning, students. Today we are going to perform the experiment of 12th biology practical, that is to study pollen germination and growth of pollen tube. For that purpose we need flower, we need slide, cover slip and these stains. First we take a slide. This is the slide. From this flower we separate a stigma which is pollinated, on which pollen grains are present. So we separate this stigma and we keep it on a slide. Stigma with slide. Then we put a drop of safranin here to make it stain. Stain it. After some time, you remove the extra stain. Extra stain is removed. You can wash it with water. But this must remain on slide. It should not remove. Then a drop of glycerin, mount it with glycerin, and then cover it with a cover slip. For covering this with cover slip, we need a needle or a forceps. This is our needle. Dip it in water and very carefully you cover it with cover slip. Now gently press the stigma, gently press, so the tissues will spread and pollen will be separated. Pollen will be separated. Now we have to remove all this stain. With the help of a blotting paper, we will remove. And after making this slide clean, we will observe this slide in microscope. Now I am showing you this slide in microscope. Now I have already fixed the slide in this microscope. This is the slide, and this is a special type of microscope. With the help of this webcam, it sends the signal to screen. So here you can see all this dark area is of stigma. This dark area is of stigma, and on this dark area of stigma, these are pollen grains. These are pollen grains. You can see the spiny texture of pollen grain. Take a close shot. Spiny structure of pollen grain, and these are the pollen tubes which are coming out. You can see the pollen tubes also. Spiny pollen grain. So here you are observing on stigma germinating pollen grains. Right? So you will draw it, figure, and you will write this experiment in your notebook. Thank you.
पीछे बाराती आगे बैंड बाजा आए दुल्हे राजा गोरी खोल दरवाजा।
दिन तेरी चाहतों में गुजरने लगे दिल गए